All right, it's my great pleasure to introduce the second keynote speaker of this conference. You should, he's not unfamiliar to any of you. Kristian Helin was the organizer of the previous 3 highly successful meetings. I had the fortune of working with him for one of them, and I really enjoyed the time working together to organize the conference. He really is no nonsense, and he has also been enforcing the time very strictly, but now he doesn't have to do it. It's my job. So Kristian is currently Chief Executive and President of the Institute of Cancer Research, London, and also the professor in epigenetics and cancer in that institute. For those of you who are not familiar with Institute of Cancer Research, it's almost 100-year-old, well, more than 100-year-old cancer institute, among the second largest cancer institution in UK, or most influential in the world among them, where he has been there since 2 years ago. Kristian has had a very productive and prolific career as a molecular biologist, as an epigenetic researcher. He started his early career by studying cell cycle and defining the mechanism of a tumor suppressor gene, RB. But he later on shifted his focus to epigenetics and was among the first to demonstrate a whole family of histone demethylases, and demonstrating that they play an instrumental role in cell differentiation and in cancer. He has played various leadership roles, including more recently as the director and chair of cancer genetic program at Memorial Sloan Kettering. And prior to that, he was founding member of was the director of the program in translational hematology and founding member of Danish Stem Cell Center, and also a director of the Biotech Research Innovation Center at University of Copenhagen, which has been in that director for almost 15 years. Kristian has really had a profound impact in both research and also in public service by serving as advisory board and the committees in various positions. And he was recognized by numerous awards, including being elected as a European Molecular Biology Organization, the Royal Danish Academy of Science and Letters, winning the Novo Nordic Prize for Outstanding Biomedical Researcher. It's interesting to note that he is also a knight awarded by the Queen of Denmark. And he's most recently elected a Fellow of the Academy of Medical Sciences in UK and a foreign member of the Royal Society. So without further ado, Kristian, welcome to the podium.
Thank you very much, Bing, for that very, very kind introduction, and thanks to the organizers, of course, Anya, Bing, and Karen for the kind invitation to come here today. So I'm the knight of the, All right.
Friends at home, I apologize.
All right, so it's better like that. Actually, it was a long discussion because I'm actually not royal at all. But my father thought I should take it. But it's something you get in Denmark. It's not something particular. If you've been a professor for a long time and had served, then you just become. So it's not a big deal. But anyway. So thank you very much. I think this is a wonderful meeting. I've really enjoyed to be here, and you can hear I have a few questions, sometimes not allowed to ask them, but I go to a lot of cancer conferences, and so what I really love here is that there's a lot of mechanistic studies, a lot of structural biology, things you try to understand what's really going on. And you have to say, well, I'm very big interest, of course, in cancer, and that's what I work on daily. There's a lot of correlative studies, and those are nice, and you need the observation, but taking them to the next step. And I think this conference is just a wonderful example of how people, dig deeply into their data and try to understand what is really going on, and reconstitutions in vitro and beautiful in vivo experiments. So, thanks a lot for organizing this beautiful conference. So my laboratory has worked for many years in what I call cancer biology, and we started working on E2F. I cloned E2F as a postdoc, and of course what you do in that stage of your career is to work on what you're good at. And so we worked for that for about 10 years in my laboratory, cell cycle control, E2F, DNA replication, apoptosis, until one of my postdocs, who was at Epigenetics at that time, went to do gene expression profiles. And he found that these E2F transcription factors regulate many of the genes that are required, of course, for DNA replication, among them the repressive complex genes. And at that time, we really didn't know much about them. It was not before 2002. We knew that PLC2 is an 8,3K27 methyltransferase, and we were working on and at that point, we have actually found that the EZH2 was overexpressed in cancer. And when the papers came out, we basically decided that the whole lab started working on chromatin, on epigenetics, and we focused on that for the last 20 years or so. What we also do in our laboratory is to work on cancer epigenetics or on cancer, and our interest is in particular in leukemia at the moment. We've also done a little work on gliomas over the years, but we are really looking into gliomas. And so why did we start working on epigenetics? Well, it was what we heard about several times in this conference, is basically the idea that epigenetic controls cellular plasticity and differentiation cues. And that, of course, cancer is a disease of differentiation and proliferation. And many cancers, in particular, some of the ones that I've been interested in working on, acute myeloid leukemia, it's so clear you get a differentiation block, and now you get basically an expansion of hematopoietic and stem and progenitor cells, which eventually, if they get other mutations, will lead to the development of acute myeloid leukemia. So I will spare you from that type of work today on the AMLs, but basically try to address some of the more mechanistic questions that we worked on the last few years, working on understanding what the role of these post-translation modifications on histones are for their impact on transcription and also their function. So I will tell you 3 short stories which we published in the recent years, so I will make them very brief. So one around the role of 8,3K27 methylation acetylation in transcription regulation and differentiation, one on the functional role of 8,3K4 trimethylation in transcription and proliferation. And then I'm not just going to mention it, the functional roles of NSD1 and 8,3K36 dimethylation in transcription differentiation, because Jenson will actually present a poster about that. It was abstract 232, and he will do that this afternoon. So I will spare you that and not take the thunder from what Janice actually could show you. And then the last part, the major part of my talk, I will try to, some studies we've done over the years to try to address the language of histone modifications, which of course is, work which I would say almost dedicated to Dave Alice's work we started a bit before, then Dave unfortunately died. But still work in progress and interested in hearing your feedback on it. I've presented a few times and I do get like wins, stands like this, but we'll see how that goes. All right, so first small story, addressing the roles of 8,3K27 methylation and acetylation. So this has been done really nicely, and we saw a talk by Dan McKay the other day, and Dan McKay and Bob Derunier has addressed it in Drosophila. And also Jörg Müller, of course, have done as well by reintroducing mutations directly into the sites which can be methylated. and try to understand, so what does that do to Drosophila development? Of course, there is the saying is that what is true for a bacteria is also true for the elephant, but it's not always the way it works. So I think it's important also to try to address that in mammalian cells. So we've been working on that for some years. And so, but first a little bit about PLC2. You heard about it. It's a very important factor for regulation of transcription. It's essential for normal development. We see these hyperactivating mutations in lymphoma and melanomas, and we see these very specific dependencies when you see mutations in the SWI/SNF complexes. So therefore, there's basically been developed EZH2 inhibitors, and they've been approved for the treatment of certain cancers. They have not worked so well as we hoped for, but that's partly to do to that these inhibitors are not really good, so they're not stable, and this could be one of the reasons. And the other reason, of course, is that what we all believe in cancer now is that you actually need the treatment with several different inhibitors at the same time, so monotherapy by and large doesn't work. So if you get to P300 and CBP, as you all know, they are co-activators transcription, they have redundant functions, they sense again for normal development. And K27 acylation in particular has been used as a mark which is enriched at active enhancers and promoters. And then we also see, again, mutations in cancer, expressed diffusion proteins, mutated in solid cancer, and again, P300 CBP inhibitors are in clinical trials as well. So there's a good combination of trying to understand these things, and actually, if you work on cancer and interested in targeting some of those proteins in cancer. So what did we do actually to address the functional role of a K27 methylation and acetylation in transcription and differentiation? Well, what happened was that Faisal Mohammed, my laboratory at the time, he developed an assay to try to introduce specific mutations in all the histone genes in mouse embryonic stem cells. And to do that, he used base editors. And what he did was to develop a PCR technology so he actually could find the clones which most mutations in. So in this way, he got to basically mutate 24 of the alleles, and we introduced the other alleles in another way. So just to give you the power of this thing is that he actually could use one oligonucleotide, and in one go, he found 22 mutated alleles in one cell line. So this is how good it can work. So now in a former postdoc in my lab, who has her own lab now, she's developed a better technique where you can use prime editing. And so now it's actually feasible to mutate these histone tails with very efficiently now. So we can start addressing that. Of course, the caveat when you do that, in this case, is that you not only mutated for lysine for its methylation, but also for acetylation. And not only do monomethylation, dimethylation, and trimethylation, but you also have to take into account whether there are different functions of mono, di, and trimethylation. So there are some caveats to doing that, and therefore it can be difficult to interpret the result. So what we found was that we can mutate that, and we basically see, as shown here, you lack the mono, di, and trimethylation, and you don't see, which is important, which is shown over here in the knockout, we don't see this huge increase in acetylation that you see by when you mutate SUC12 or one of the PLC2 complexes. There you also see this huge upregulation of acetylation because you still have lysine, and part of what the methylation does is actually prevent acetylation. So it has been speculated that much of the function of K27 methylation is basically to block acetylation so you don't get gene activation. So what our results showed, I'm not going to go through the data because we published that you see a couple of years ago, is basically what our results show that this pan mutant, it basically replicates the polycomb knockout phenotype in early differentiation. This is done in ES cells and then differentiated into embryo bodies. But also what we could show is that K27 acetylation is not required for transcription. And so we got a lot of pushback on that because people, they love K27 acetylation. There's nothing wrong with that. It doesn't say that K27 acetylation is not important. It just says that it's not essential and that there are many other sites which are hit by P300 and CBP, potentially which are redundant to A3K27 acetylation. And finally, I think what was important, and then hopefully we'll get a lot of people to do that, it's feasible to mutate all the histone genes for functional studies so we can get above this thing, what the correlation. We know many of other proteins which are actually methylated or acetylated or something like that by these proteins, and they may actually be important, but at least not in this thing for early development. We can replicate the phenotype very much similar to what has been shown previously in Drosophila. So the other story I will tell you briefly is one about H3K4 methylation in transcription and cancer as well. And we got a beautiful talk also earlier in this meeting from Drosophila, where we saw that it actually affected transcription, which is something which has been discussed for years as well. Is it something coming after, or is it something which is actually involved in transcriptional regulation? So if you look at H3K4 methylation, we have the monodiene trimethylation, and this is typically what you see for the genome. It's not as specific as people would like it to be. It's sitting very specifically at certain sites. And this might, by and large, also be due to the antibodies used, because as we know, we go back to literature, and many of them were not as specific as we thought. But one thing which is for sure, and I think we all can agree on, is that trimethylation of K4 is a very, very good mark of CPD islands, and the higher it is, also of active genes. So it sits there, right there, and if you didn't actually find a promoter there, that's because you haven't annotated the transcription start size well enough, because for sure it is sitting there where you have transcription start size by and large. And so what is that methylated by? By a big complex here called basically it's called Compass, and we have Mr. Compass sitting over here, who's giving a talk later today, who has been pioneering a lot of work around defining that complex and his role in transcriptional regulation. And the complex have different types of subunits. So they have these core components, and then they have a set one on MLL component. And there are basically 3 different types of complexes, and you can recognize those with these specific subunits which are there, which distinguish the different complexes. There's been speculation what they do. They have special role, and I will let Ali tell you more about their special role in different types of regulation. But in particular, one of them here has been suggested to be enhancer-specific, and there's been discussion whether monomethylation is required or not, and I think the verdict is right now is probably not required for enhancer activity. Anyway, so we started working on that, and this is the role in cancer. I'm not going to tell you about that, but we started to actually asking the question, which is addressed up there, is basically the idea is H3K4 has been reported important for transcriptional initiation, consistency, memory, as well as post-transcriptional events. And others have suggested that K4 methylation is not important for transcription regulation at all. I mean, that's a good starting point, isn't it? So now that's really a good conflict there. There are papers about that as well. And of course, also cause-effect relationship. So how does it work? Anyway, so what we did was basically relatively simple. And I have to say we're a little bit lucky here as well. So what we did was to try to make inducible degradation of some of the core subunits of all the K4 trimethylated residues. And this is DPY30. We also have one with RBP5, and we use different tags, and we show very similar results with those. So what you see here is DPY30, which is one of the core subunits degraded. This is an auxin system. And what you see, which we're surprised about, is that K4 trimethylation is wiped out. I mean, we knew it was probably not the most stable thing in the world. This is ES cells. We haven't done that in other cells, so that's a caveat. But in ES cells, it's turned over really, really rapidly. So within 3 hours or 2 hours, depending on your sensitivity to the assay, you basically lost all K4 trimethylation. And the lucky thing here is perhaps to say, well, it took much longer for and monomethylation. It means that if you do the early time points, you have an idea that you can actually distinguish the different functions of K4 tri from what di and monomethylation they do, because you can map them genome-wide, which Juan in my lab did. So, as I said, we published this last year in this paper, and basically what the paper showed was the K4 trimethylation. It regulates RNA polymerase II, promotes a proximal pause release. And the mechanism, not 100% sure, but what we did see was that there was a potential function for integrator complex in that. We lost that by K4 trimethylation. And so something we've been discussing with Karen, sitting over here, is that what is the specific function of that? The verdict is there, but I think we saw at least many of the results which were compatible with that the integrator had in these short-term assays similar function as losing K4 tri. I think the biggest thing was I thought, at least when I saw that, is how unstable the K4 trimethylation really is. And it's not much of an epigenetic marker, at least not in ES cells, when it's turned over so fast. And why is it turned over so fast? Well, because the KDMs, as you saw here, KDM5A and B, they're sitting there all the time and turning it over, which most likely is important for regulating transcriptional bursting. So you actually have a feedback system here, so you downregulate K4 trimethylation all the time. So we also showed some other things. This is always a difficult thing to say, but at least our results, they suggested that K4 trimethylation is not required for transcription initiation, but it is required for the recruitment of the integrator complex that is also required, at least in our studies, for the promoter proximal pause release. And there are a lot of studies that from Karen Adelman's lab and, of course, from Ramin Shigatar's laboratory, who knows much more about integrator than I do myself. Right, so just a bit summary here before I get to the next story. So basically the functional role of histone modification, they are important. Wow, that's number one, right? Because this has been, something we've discussed for a while. Some of them are important. It doesn't say that all of them are important, but some of them actually do something, which is quite important to know. So K27 methylation explains the functional role of PRC2 in transcription differentiation. K27 acetylation is a marker of enhancers, but not essential for enhancer activity. Remind you, it doesn't mean it doesn't have a role in enhancer activity. It just says it's not essential for it. And then trimethylation is essential for RNA pol2, post-release, and transcription. And what Jen is going to show is that NSD1 has a catalytic independent function in regulating enhancer activity and a catalytic dependent role in development, which is interesting as well. You do not necessarily need the same activity for regulating the same thing. And so why are we able to do this? And that we've seen a lot of these in this meeting. I've seen developing of new technologies actually allows us to ask some of the questions we addressed before much better than we've done before, and therefore we get more temporal resolution. We have better assays to actually understand what is going on, and now we can also, because of CRISPR, that wonderful technology, we can actually do reasonably good genetics in mammalian and human cells. Good. Let me go to the next part. So this paper is something we sat out here, what is it, early 10, 15 years ago, discussing this paper and the importance of the language of histone modification, the so-called histone code. And this was first, at least to my opinion, what I know, basically suggested in this paper from Brian Strahl and Dave Alice in Nature, where they basically suggested that there was a language where these histone modifications, they actually were determinant for transcription. And that, of course, got a lot of smoke because, it was known that acetylation seemed to be promiscuous, so how could that be a language? And so basically it was formulated perhaps even stronger in this review by Thomas Ginowine and Dave Alice, where they said, well, histone modification can generate synergistic or antagonistic affinities for chromatin-associated proteins, thereby dictating the chromatin state. The combinatorial nature of histone modification reveals a histone code in which information can be integrated into unique downstream effects. I think most of us, ascribe to that type of talk. It's not bad, right? And I think we think about it like that as well. The question is, how much is the code? How much is there degeneration in the code? And can we substitute one thing from another? So we try to address that. This so-called histone code hypothesis, namely that histone modification have unique effects on transcriptional activity. And this has, of course, been mostly done by locus-specific and genome-wide correlative studies, which broadly have characterized modification as either active or repressive. And you remember first time they got out with a 3K4 chip, and Tony Kouzarides was talking about his result. He said, well, are these genes active or not active? And he said, well, there's 50-50 chance, because half of them we learned later, so on bivalent genes, and they were not active. So one modification by itself does not necessarily tell you whether a gene is active or not. All right, so there is a potential redundancy between the different histone modifications, as we know, and if the transcriptional outcome is the same, repressive or activating, one could ask the question, why are there so many different histone modifications? It doesn't make sense, right? Why would we need them? And why does it matter which histone modification directs the transcriptional outcome? So a former graduate student in my lab, Lenny Hill, who started working on that 5-6 years ago, and we're still working on it, so one day we'll get out. Many problems on the way, have 2 moves and whatever, has not helped for that. So what did we do? Well, so what Lynn did was try to focus on what we call repressive modifications, so K27 tri, K9 tri, and K36 tri. And we can have a little discussion about what K36 tri does. It's not repressive as the others, but it does seem to be repressive. And what we know about them, well, they had distinct areas of the chromosomes where they are located. And we take K27 trimethylation, which is totally catalyzed by PLC2, CBD island promoter regions, transcription silent genes, and they maintain transcription repression. When you get K9 trimethylation, it's mainly catalyzed by the SUVA 39H1 and 2 and ZDB1, gene poor, heterochromatic regions, entegenic regions, and repetitive DNA elements, repressive through recruitment of HP1 and DNA methylation. And then finally, K36 trimethylation, which is actually associated with active genes, catalyzed by ZD2, the only enzyme located in gene bodies at active genes with increased abundance towards the 3' end. We define it as a repressive because it leads to DNA methylation. And it's been suggested that it actually prevents transcription initiation at cryptic start sites within the gene body, and it leads to this DNA methylation over the transcription start site. There are results showing that it can be repressive. So K9 trimethylation, K27, K36 are all considered repressive modifications found in different genomic contexts. And so these all seem to exert a repressive function, but are they unique, we ask ourselves? Or can K9, K36 substitute the genome-wide pattern of K27 trimethylation with similar functional effect on transcription? So how do you do that study? That's the question, right? So how can we address this question? There are two ways we can do that. There's this one, which has been used, going back to what Mark Patashner did, showed many, many years ago, that transcriptional regulation is modular. You have a DNA binding domain, you have a transactivation domain, and you can separate the two, and you can put another transactivator on, and they bind to the DNA. This we can do these days using a DCAS9 system. DCAS9 in with a guide RNA, and then you can recruit it to where you want. Of course, the disadvantage is that you become locus specific, and you can only study relatively few genes at a time, and you don't put it into the natural chromosomal context. So what we did instead was basically expand on this principle by hijacking a part of the PLC2 complex as the recruiter module for actually putting other activities in exactly where K27 or the PLC2 complex sit. So in this way, we could actually have a robust assay, and we could put whatever activity we wanted on a PLC2 target gene if we knocked out the endogenous PLC2 complex. So that's what we did. So we published a couple of papers some years ago where we showed that the PLC2 complex is indeed modular. We're not the only lab who has shown that, but I think it was more clearly shown in these results here. So this is the PLC2 complex. So this is the catalytic activity, and there's this VEFS domain up here of SUSI12, which binds to EZH1 and ED. And there's this SUSI12 N-terminus one, which binds to other proteins. And this is what leads to the recruitments to specific sites in the chromatin. So what happens if you delete this part of the protein? You actually retain catalytic activity, and now it's distributed throughout the genome, but it's not localized anymore. So the SUSI12 N-terminus is what actually focuses on the CPD island. But what is important for our study is that you can take this, just what we call the N-terminus of SUSI12, and it localizes exactly to the same place on the peak sites of the PLC2 target genes in embryonic stem cells. It's not the same level for some reason, perhaps there are other, but it actually, what it is important is that it's specifically localized there. So what we could do then is basically use this part, and we could put different catalytic domains on there. And so in this way, now we can put at will what we want on the right side where you otherwise would see K27 trimethylation. And we did that, and here it's shown here for SUVA 39H1 and for ZD2. We took the catalytic domain, so we also called the N-terminus for the VEFS domain. And so in this way, we tried to test that. So they are expressed here, and this is then how they are loaded onto the PLC2 target sites. So you see here, there's clearly a reduction, which is perhaps important to think about. So there's a reduction here of the level of SZD2, and this is then SUVA 39H2. All right, so the question is, of course, when you do that, does it result to patterns of K36 tri and K9 methylation respectively at PLC2 target sites which actually resemble H3K27? I wouldn't tell you if it didn't. I can promise you that much. Anyway, so this is the way it works. So you look at specific target genes, and this is like looking at what K36 tri does. Let me just tell you what goes on here. So these are wild-type mouse embryonic stem cells. When you knock out SUSI12 in those, you lose all K27 methylation activity in these cells. So they're just blanked out. All right, so this is K4. You see in the same cells, you see some increase in K4 methylation because you see increased transcription as well, and this is the Hox locus. So now you look at K36 tri, blank, blank. You express this ZD2 fusion protein together with the SUSI12. Now you see K36 exactly where you see the K27 trimethylation, and if you have a mutant, you don't see anything. All right, that's all good. So the question is, of course, what is the level you're getting here? Are these levels physiologically relevant, right? Or do we get too little because we have, too little of K36 bound there, or the ZD2? So we looked at that, and this is a good gene to look at this, because here you have, again, a similar study on two other genes. One is an active gene, and one is repressed by K27 tri. When you remove it, you remove K27 tri. Now you get K36 when you do that. And these are the endogenous genes which are always expressed. And you see the levels you get here are very similar to the level of K36 tri you see on an endogenous gene. So it seems to be physiologically relevant for K36 genes. We see over here, the peak is exactly where you see PRC2 being located in K27 tri, and we see a linear relationship about how much K27 tri that is in wild-type cells versus what we express. So recapitulating really well. Okay, so the system seems to work. We drive K36 tri methylation deposition and largely recapitulates the genome-wide profile of K27 tri methylation. And so, what I did there, yeah, well, that's good. We could do the same for K9 trimethylation, which is shown here. So basically, same idea. We have SUVA39H2 here, and we have a catalytic mutant. Other locus here, K4. So this is what you see at K9. Some of the peaks are there all the way through. Now we put the fusion protein, and now you see you put K9 trimethylation at the spots where you have K27 trimethylation. So some of you probably saw that there's a bit hole here. There's a little hole there, and there's a little hole there. And when you do that genome-wide, so this is where K4 trimethylation is in wild-type cells. So you get the modification of K9 trimethylation all over. The problem is when you have high levels of K4 trimethylation, it actually prevents K9 trimethylation to be sit at the very same site. And we know that from the literature, you never find K9 and K4 at the same site. So it prevents K9 there. All right, so we've generated these systems, so now we can see what happens in transcription. And so the question is, of course, obvious. Can K9 and K36 substitute for K27 trimethylation in regulating transcription? I know some of you have seen this before, otherwise I will ask you, what do you think happens? For those who have not seen it before, you can answer, what happens? Does it work? No. There was a no. All right, let's try. All right, so this is then, so these are we're looking at gene expression changes in SUSI12 knockout cells. You knock out SUSI12, you see a number of genes going up, you put back SUSI12 so you can rescue a good number of them. Now you put in those cells, you have this basically the fusion protein giving rise to K36 trimethylation. Not very good, right? Much better here with K9 trimethylation. So it's not interesting if I just say, oh, this is a result, if we don't try to go under the result and try to see what's going on. So it's basically, when we quantify that, so, depending on how you do it, but we basically rescue the expression of 75% of the gene. When you put in by just the N-terminal of SUSI12 doesn't rescue at all. When you put in with a ZD2, 3.5% are rescued. So it's pretty terrible, right? But that's the number. There's some reason for that as well. When we put in H3K9, we get like rescue about 25% of the gene, which is not bad at all, not bad. And the mutant doesn't do anything. And interesting, and probably as expected, we can't basically rescue differentiation defects we see in ES cells lacking the PRC2 complex. All right, so they don't seem to have the same repressive activity, and we try to ask the question, can this be explained by the chromatin environment? And we ask these two questions, which elements of the chromatin environment may explain the lack of repression by K36 tri, and very similar for K9 tri, whereas there we had something positive to look at, which was a good thing. All right, first of all, K36 trimethylation and DNA methylation. This is important to know about because we think this is the repressive mechanism. So it's important when you see that, when you see K36 trimethylation, the position is efficiently deposited at PRC2, K36 tri is efficiently deposited at PRC2 regulated target genes. K36 trimethylation repression is mediated through DNA methylation, but endogenous K36 trimethylation and K4 trimethylation do not overlap. So the question is that there are multiple studies which have shown that K4 trimethylation protects promoters from de novo DNA methylation. It basically does not allow DMT3A to bind to those promoters, so you can't set up DNA methylation. So we looked a little bit into that, and we looked at DNA methylation patterns here. I'll go a little faster. So what we did is that we get a feeble increase in DNA methylation on promoters which have K36 trimethylation. And interestingly, this 3.5% where we see some level of repression are the ones which have lowest levels of K4 trimethylation and highest levels of DNA methylation, suggesting that we have a protective mechanism for the repression which is mediated by K4 trimethylation on those promoters, even though that we put in high levels of K36 trimethylation. So that was one result. The other thing was that when we looked at basically showing that the low levels of DNA methylation may explain the lack of repression mediated by K36 trimethylation at the PRC2 target genes, the question is what happens in the other chimeric protein. So we found that K9 trimethylation was not efficiently enriched at a subset of K4 tri positive PRC2 target genes. And the question is, could we actually functionally test whether this was important for preventing methylation and also therefore transcription repression? So what we used was a mutant, basically we made cell lines with SUSI12 knockout cell lines, and we used this depletion system which we published last year, which I just told you about before, where we have RBP5, which is basically fused to FKBP12, and so introduce that into these SUSI12 knockout cells. So if we treat them with DTAC13, we can then downregulate RBP5, and we lose all H3K4 trimethylation within a couple of hours. And so we did that type of experiment in these cells, and what happens? Well, so this is first what happens with K4 trimethylation. So we treat with DTAC, you basically lose K4 trimethylation. Now we have expressed the fusion protein, basis SUSI12 N-terminus with SU39H2. And so this is the level we see here. Now we delete basically K4 trimethylation, you see this increase, and you see an increase in K9 trimethylation. So there's a protective mechanism against being K9 trimethylation. And we see twice as many genes which are now repressed. So basically now we're up to 50%, and now we'll start talking business. Anyway, so the model we're ending up with here is basically there is this hierarchy and crosstalk between histone modifications and their corresponding enzymes which prevent K9 trimethylation and K36 trimethylation to repress PRC2 target genes. So what we see here is that K4 tri is dominant over K9 tri, but if we knock it out, you get more K9 trimethylation, and similarly for K4 trimethylation, it seems to be dominant over DNA methylation, although I haven't showed the results that this actually leads to increased methylation. So basically it highlights the critical role of K4 trimethylation in safeguarding CPD island promoters to ensure appropriate transcription regulation. And just to conclude, so in summary, what we've done is basically we've shown that K27 tri, K9 tri, K36 tri, these are all classified as repressive modifications. However, they are not interchangeable because of the chromatin context, even though they can all repress transcription. Crosstalk between histone modification ensures incompatibility. Modifications are at spatial separation. And the crosstalk is a potential mechanism ensuring precise regulation of gene expression during differentiation and development. And that's why they might be so important, and this is part of the mechanism by which they work. And I would say the conclusion would probably be that our work support what Dave Alice suggested 24 years ago in a Nature paper, and it emphasizes the importance that we probably all think but we try to address in this study that the chromatin context is very important, and these modifications do have diverse functions in regulating transcription repression. So with that, I will just end my talk by acknowledging the people that did it, particularly Aditya Sankar and Faizan Mohammed on the K27 methylation acetylation. Chenson, you're going to meet later at poster 232. And then Hua on the K4 trimethylation, Lin and Robin on the last part of our talk with some help from others in the laboratory, our collaborators and our funding. And with that, I will be very happy to take questions if we have time.
Great. We have time for a number of questions. First one.
Well, separate the argument that goes against the histone code, as you mentioned initially, monomethylation and acetylation on enhancers don't play a role in enhancer function.
Yeah.
And that's where everything started. In yeast, when you knock out all the lysine residues, there is no effect on viability, so histone modification. So I would have thought in the experiments that you would do, you actually try to differentiate between the context of the enzyme and modification, right? So if you bring a catalytically dead PRC2 to where PRC2 is supposed to go, what happens then? You put a bunch of marks, different things happens, but you're not considering the context of fully put together PRC2, but that is not catalytically functional.
But that's been done not by my laboratory, but by other laboratories, and you don't rescue. You don't rescue it by putting a catalytic inactive mutant.
On K27 methylation.
Yeah. But also, Ali, we know we have inhibitors specific for the catalysis of PRC2, which have actually been improved for the treatment. So we know the catalytic function is important. Otherwise, they wouldn't work.
But none of those inhibitors are doing much in the clinic, right?
Well, they still have been improved. I would just say they still have been improved.
But doesn't mean that they're doing what they're supposed to be doing. So I think you should consider the context of the enzyme a lot more than the modification. That's all I wanted to point.
No, I understand very well, but the inhibitors do not degrade the enzyme or the PRC2 complex. Yeah.
Is it me? Okay. Very, 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 Kristian. Great talk. One of the things that I was wondering while I was listening to you is sometimes, I don't know if you remember, when we put K36M, we had a spread of the trimethylation and we diluted PRC1. How about if you have way more K36 tri? You have enough of DNA methyltransferases to kind of coat it? Is this, so I really would like to see the DNA. I know you showed hypomethylation, but is it due because you're having the K4, or is it due to the level of the expression of those DNA methyltransferases?
Yeah, so that's the experiment we didn't do. I mean, we can do that in our cell line where we knock down all K4 tri to see whether we increase DNA methylation and therefore get transcription repression. They are a little bit tricky, those experiments, because we can't keep the cells in culture more than 24 hours because they die. Yeah, no, I mean, yeah, we haven't done that. It's DMT3A, but anyway, yes.
36.3 is more DMT3 than 3A.
Okay. Yeah.
Okay. I was curious, with the K4 trimethylation blocking the K9, I think this makes sense because of the close proximity and affecting the catalytic activity. Is the K4 trimethylation usually at those sites, or is that a consequence of knocking out PRC2?
I mean, what you normally have is, so I would say all CPD islands have some level of K4 trimethylation. Those that are repressed by polycomb, really repressed, they have very low levels, but they have some. This is the bivalency, if you want. When you then remove the PRC2 complex, you see some upregulation of K4 tri on those target genes.
Yeah, I'm just curious whether the K9 would have been able to compensate if that state wasn't already being maintained.
Yeah, so K9 trimethylation, you rarely find a transcription start site. So I think what is going on there, and that's what I'm trying to say, is that where you see K9 coming up are actually the ones which have low K4 trimethylation. That's where you get the K9 trimethylation. And when we then remove K4 trimethylation, now you see an increase of genes which are positive for K9.
And then I was most curious about the fact that the boundaries stay consistent when you use the fusion. So what do you think is maintaining those boundaries?
In fact, K9 tri spreads more than K27 tri in our experiments. I don't know for K36 tri, that we were a bit surprised about too. Because I did, when you do these experiments, you don't know what you get, right? But this beautiful spread of K36 tri, I did not expect that.
Thank you so much.
Hi, Kristian, here.
Yeah.
Here.
Okay.
Yeah. I have a question about K9 methylation. Whether you see where the heterochromatin protein 1 goes there.
Yeah.
That's one question. And the second question is, so K36 trimethylation, I thought that it's in part the set is stimulated by interaction with RNA polymerase to get to the trimethyl state.
Yeah.
Do you see pull2 actually after you recruit the K36 to that loci? I mean, if you would look at the distribution of...
So you're asking, can it work the other way around?
Yeah. So I mean, are you recruiting the factors actually of yeah?
Yeah. So we don't see an increase in transcription. That's your own. We haven't looked at RNA pol2, so that we can answer. So we are doing experiments. So we did send it out on the review and so on. So we are doing an experiment with HP1 to see, what happens when we put HP1 there instead of the other, and it could be part of the things. Because I think one of the problems is that they're not, some may know better, but they're not very good HP1 chip grade antibodies, so it's very difficult to actually look at HP1 recruitment.
Yeah, a quick question. Probably I missed this. I just wonder, if using DNA methylation is the cause, have you tried bringing DNMT to the loci, and does this rescue the differentiation? Yeah.
So we actually made fusion proteins with this thing. It was the first thing we did. We made that so one was working on the DMT3A, 3B, and put that together with the N terminus of SUSI12 to see whether that could repress, and that didn't work. But this was. Maybe we did not, let's say, investigate it well enough to say, well, know the reasons why it didn't work. But we did try to do that.
Dan, okay, have the last question.
Kristian, beautiful talk. It was interesting to see that when you knock down K4 methyl, you boosted the ability of K9 methyl to repress. Yeah. Is it possible to wash out the degradation and ask if the K9 methyl repression persists?
Yes, we haven't done that. That's a good experiment. It's definitely possible. These are reversible, fully reversible. So yes, good experiment.
Great. Let's give Kristian another round of applause for a beautiful presentation.